Hello everyone, thank you for coming. I'm James Anton Giovanni.
My name is Rita.
I'm Stephanie Friedrichsen.
And I'm Kathy Lopez, and our presentation is going to be on a parasite called Heminiscus colani and the effects of multiple infections. Our mentor is Caitlin Fong, and our PI is Armand M. Perez. So when you look at this picture, what do you guys see? Barnacles. Correct, they are barnacles. But what we happen to see is that up to 90% of these barnacles may be infected with a parasite. Parasites make up to 60% of the animal population, which is a large percentage. However, because they require a host in order for survival, it's really hard to research them in their natural habitat and the living conditions due to the fact that you have to kill the host in order to get the parasite.
So the parasite that we are studying is this one right here. It's called Heminiscus colani. It is also known as a castrator parasite. Parasite castration basically means that when the parasite enters its host, it has the ability to stop its host's reproduction system. So this particular parasite will enter a barnacle, the left picture over there, the Cephalothalmus physis. When it enters this host, it will attach to the ovaries, it will suck out the ovarian fluid, and then it'll use that as energy for its own reproduction purposes. Now, how can you tell whether your host is infected or not? On the left-hand side, we have a barnacle that is not infected. On the right-hand side, we have a barnacle that is infected. How you can tell is you see where she's pointing, you have a reddish-orange blob. That is the parasite. And an interesting fact to know is that the parasite and the barnacle are about the same weight.
So we have some questions we would like to test. First of all, we tested. First of all, we believe that as percent host infection increase in a population, we would also see the percent of multiple infections within that sample size increase as well. We believe that larger barnacles would make optimal hosts because we think those would have been the ones that had the most reproductive fluid and reproductive tissue for the parasite to utilize. And in multiple infection cases, we thought that there would be competition in that one parasite would be dominant, producing more net eggs of the total, whereas the other one would be submissive, producing less.
So our research requires to do some field work such as collecting barnacles from their natural environment. The two main points that we collected were coal oil point and canvas point, which are the rocky intertidal zones. For method, we use dissecting microscope, which provide us a chance to have a closer look on our species, which are the barnacles on top and the parasite which is on the bottom, right here. About the procedure, we first measure out the size of the barnacles to the nearest quarter of the millimeter, and we try to see if is there any chance of infection. So once we took out the parasite, we're gonna count the number of parasite per barnacle and also the number of eggs each parasite produced. The following graphs were made based on our data collection.
So our first graph emphasizes that indeed as percent of hosts infected in a population increases, we do see a percent of multiple infection also increase. This is a y equals mx plus b graph, and on the x-axis we have our percent of hosts infected. For every 10% of hosts infected in a population, we see about a 1.7% of multiple infection increase. An easier way to think about this is for every 100% host infection in a population or sample size, we see about a 17% of multiple infection increase. Our sample size here was pulled from about 12,457 infections.
Next, what we wanted to see if infection correlated to the size of the host. And there's a lot going on here, but what we see down here is the length of the barnacle in the size of millimeters. And then over here on the left, we see the percentage of probability infected, meaning how many parasites infected a barnacle. And this scales up to 50%, and it'll be represented by the dashed line here. On the right side, we have the percentage of probability of reproductive tissue that each barnacle has, and that's on a scale of 12%, and it will be defined by the black line. Now, what we want to see here, and the trend that we see here, is that the optimal size for the parasite to infect a barnacle seems to be 4 millimeters. Both of the maximas for both probability of infected and probability of reproductive tissue tend to be here. And that will correlate to the fact that because this size of barnacle tends to have more reproductive tissues.
So looking at infection in a population, as Kathy said, we found out that that 3 to 5 millimeter range is optimal for the barnacle size for the parasites to utilize their reproductive tissues. So the barnacles we see are hermaphroditic. They start out as male. They gain their reproductive organs around that 3 millimeter mark, losing their reproductive tissue around that 5 millimeter mark. So this would be the optimal size that would be infected because they're the ones that have the ovarian fluids the parasite can utilize for its own egg production. So looking at this, we noticed that most infections were in that 3 to 5 millimeter range, that optimal range. And looking at that, intuitively thinking it does make sense that that's where we would see most of our multiple infections as well. And our sample size here again was 12,457 infections.
So we do know that multiple infections occur, but what we also want to know is whether the net number of parasite eggs is less when there's 2 or more parasites within one barnacle, or we usually call it multiple infection. So we compare the number of parasite eggs that came from the single infection when you have one parasite within one barnacle, or multiple infection when you have more than 2 parasites within one barnacle. So for this graph, we have down the axis, we have the type of infection, which is either single or multiple infection. Y-axis, we have the number of parasite eggs, the total number of parasite eggs produced. But our sample size is 53 barnacles, which is very large. So as you can see right here, the number of parasite eggs that came from single infection, which is around 84 eggs. From multiple infection, it's roughly 100 eggs. And we see little to no difference between those two numbers. So our group conclusion was that for this particular barnacle, whether it's had multiple infection or single infection, they have roughly the same number of parasite eggs.
So in this graph here, we're looking at the percent of parasite eggs in correlation to the parasite characteristics. So because a host can have multiple infections, we will have dominant and submissive. What that means is that when you have 2 parasites within one host, one parasite will produce more eggs than the other. The parasite that produces the more eggs is the dominant parasite. So the submissive one produces less eggs. So what we can see here is that we have a 70-30 dominant to submissive relationship.
So some conclusions that we made is that indeed as the more hosts that are infected within a population, we will see more multiple infections. The intermediate host size happen to be the more optimal host because they are the ones that have the reproductive tissue that the parasite can utilize, which is that 4 millimeter mark. So the number of infections does not affect the net egg production, meaning that rather we have multiple or single infections within a population, the net eggs produced by the parasites in the 2 types of infection cases is relatively equivalent. And our data does suggest that in multiple infection cases, we do have competition and one parasite will be dominant, producing around more than the submissive parasite. A good example of this, let's look at 2 parasites in one barnacle. One parasite will be dominant, producing about 70 eggs, whereas the other one will be submissive, producing about 30 eggs.
Now, this research can go in many ways, but what we can probably focus on next time would be host location correlated to infection rate, meaning because the barnacles are on an intertidal zone, which means they're not always exposed to water, so we wanted to see if the barnacles that were longer exposed within the ocean versus not, and see if infection rate changes. And as well to see if other species within this habitat are also infected by this parasite. Thank you.